Hello everyone, I'm Mr. Furlong and you're watching Chemical Reactions and Enzymes. Today we're going to take a look at chemical reactions and enzymes. Without these two things, we would not be here today. Why do I say that? Well, because for a cell to stay alive, it needs to have thousands of chemical reactions taking place every second. And in order for all those chemical reactions to take place, we have to have enzymes, like this enzyme here. And I know what you're thinking. That looks a lot like the proteins we saw in the last video, and you would be right. Enzymes are a type of protein, and we're going to talk more about these chemical reactions and proteins here. Chemical reactions are a process in which one set of chemicals, the reactants, change into another set of chemicals, the products. There are lots of different kinds of chemical reactions, but we're going to only focus on two this year. These are the two most common ones that happen inside of cells. The first type are called synthesis reaction. Now this comes from the root word to synthesize, which means to make or to build. So a synthesis reaction is actually a building up process. The actual name that scientists call it is a dehydration synthesis reaction. Let's take a look at why that is. Let's say that we have a glucose molecule, C6H12O6, and we combine that with a fructose molecule, another monosaccharide. What we get is a disaccharide, and notice at the end we make a molecule of water. So in essence, one molecule of water is released from this reaction. Or in other words, it's dehydrating, it's losing some water. The second type of chemical reaction is called a decomposition reaction. Now you've heard of the word decompose before. That means to break down, and that's exactly what's happening. We're taking larger organic molecules and breaking them down into smaller organic molecules. The actual name for this is called a hydrolysis reaction, because in a hydrolysis reaction you take a water molecule and you split it. Hydro means water, lysis means to split. So in this example, if we were to take a sucrose molecule, that's a disaccharide, and take a water molecule, split it, we get two monosaccharides. In order for these chemical reactions to take place, we need two things. One is energy. Specifically, we call the energy needed to start a chemical reaction as activation energy, or the amount of energy needed to activate that chemical reaction. So think of activation energy like this. On this graph, here I have, let's say I have a big old boulder, and I want to get it from here to here. Now, of course, I can roll that down the hill, but the problem is I have another hill that I have to get that over. So I need to apply a force. There's me applying a force. And of course, my brute strength, I'm able to do this actually pretty easily and get it to roll down the hill. But that does take quite a bit of energy. If I use an enzyme, well, look at the size of the hill that I have to get that over. It's a lot smaller. And so the use of an enzyme really helps me to get more reactions going in a shorter amount of time. Enzymes, we could say, speeds up chemical reactions then. Some of these reactions might take a long time to take place, and we need to have thousands of these reactions occurring every second. So enzymes are going to be very helpful. It lowers the amount of activation energy that we need to get these reactions going. So think about that. We can get more chemical reactions, and it doesn't take as much energy to get those reactions started. These enzymes are reusable. They can be used over and over and over again. Most of these enzymes are made up of proteins. The thing about these enzymes, though, is they can only catalyze one type of a reaction. So one enzyme can't do all the different reactions that we need. We need a different enzyme for each different reaction. And in a human, that's roughly around 2,600 different enzymes to catalyze all the different types of reactions that are going on. Scientists often refer to this as the lock and key method. We have keys for all kinds of things. We have a key for our car. We have a key for our house. I have a key for my classroom, and I have a key to get these paper towels changed. Each one of them looks different from one another. Well, that's kind of how enzymes work. Every reaction is slightly different. So I need a different enzyme to catalyze that reaction. So let's take a look at how this is going to work. Here I have just a simple geometric representation of an enzyme, and any substance that an enzyme acts on is known as a substrate. So that's just a generic term. Notice how the enzyme, it has this nice three-dimensional shape, but there's one part that, right here, I'm going to highlight this, that looks different from the rest of the enzyme. That is known as the active site. Now that has a very specific shape to it, and if you take a careful look, the shape of the substrate is complementary to the shape of the active site. So the enzyme can grab that substrate and it'll fit right in the active site. And when that happens, we have what is known as an enzyme substrate complex. Now, if this substrate was any other shape, it would not be able to fit in that active site, and so this enzyme would not be able to work on that. This is what makes it specific to the reaction. Then the chemical reaction takes place while the substrate is within the active site, and so let's say that the chemical reaction occurs. And now what we have is an enzyme, and notice the enzyme has remained unchanged. The shape of the active site is still the same as it was before, but now I have two products. Well, we're going to take a small amount of hydrogen peroxide, which is H2O2, and we're going to add an enzyme to it. Now, hydrogen peroxide is a poison. We do produce hydrogen peroxide. We have various cells that will produce it, but sometimes it builds up as a byproduct and we need to get rid of it. So we have enzymes to break it down. Now, the enzyme that we're going to be using tomorrow is called the catalase enzyme, and a very good source of catalase enzyme comes from beef liver. And it's pretty inexpensive to get as well. And when we combine a small piece of beef liver to the hydrogen peroxide, well, it's going to break that H2O2 into oxygen and water, which is a pretty fascinating thing. So we're going to talk more about that tomorrow. We're going to see these reactions taking place, and I can't wait to get started. See you then.